All right, so here's our second part in the genomics lecture. Keep in mind what we talked about in the first one. We were looking at what we can do with genomics, the physical map, the genetic map, and just, again, the idea that we are really scratching the surface of this topic and this field. So much opportunity, so much new information to be explored. A lot of those things we think about or see in the movies that we kind of see as science fiction, futuristic, some of those things are actually happening, and we're doing them. Not as easy as it appears in the movie, but some of that is reality in today's life. And the exciting thing is, who knows where we'll be in 10 years. We'll be there very quickly. 10 years goes by pretty darn fast, and all of a sudden, like, wow, I remember hearing about this stuff in class, and now all of a sudden, what do we have available in regards to genomics? Okay, so the last lecture we left off with looking at human ancestry, just talking about all of us share common ancestors. If we go back far enough, when we look at the evolutionary tree, we will see we all are related. So we'll definitely come back to this and revisit this in our evolution unit, spend a lot more time talking about how evolution works, the process, but the big thing is, I want you guys to remember genomics is a supporting line of evidence that helps us understand evolution better, and it confirms what the fossil record shows and what the anatomy shows and the biogeography and all these other lines of evidence. Genomics is just one more line of support for this concept or this idea of common ancestry. So, okay, so Human Genome Project didn't just start back in 1990. The history of the genome has to go back to 1865. Gentleman right there with the pea plants. Hopefully he looks familiar. Gregor Mendel. His foundation of genetic information is what helps us today when we're working on the Genome Project and trying to understand genomics. Erwin Chargaff's information in the 1940s. Look it up. What did Chargaff contribute? In 1952, there was a young woman who took an X-ray of DNA. Who was that scientist? And then in 1953, we have Watson and his lab partner, his collaborator, scientific partner. Who was that guy? So I want you guys to fill in these blanks because you're going to see this again. So big hint to you. Make sure you know the people involved in this timeline and what their contribution was to science, and then how that contributes to the field of genomics today. Okay, but it doesn't just end in 1953. We go into 1961. This is when we really started understanding the protein synthesis process. DNA transcribes to RNA and then translates to an amino acid. We didn't get a handle on that until the 60s. Early 60s is when this was discovered. So I completely understand that's 50 plus years ago, but when you look at it in the big picture of science, it's not that long ago, and it's a complex process, which is why we still don't understand it completely. We have a great base, a great foundation of knowledge, but there's so much more still to be learned. DNA sequencing didn't start happening until the 70s. It wasn't until the mid-80s when that sequencing was being automated. Think about when computers really started ramping up. I mean, most people today, especially younger folks, if you grew up with a computer, it's always been there. Computers weren't available in homes the way they are today. And when we were back in the 60s, 70s, 80s, even into the early 90s, everybody didn't have a laptop and an iPad and a cell phone and a smart book and all these other things. So as that technology came online, genetics and genomics advanced using the technology. So it worked beautifully. So as new technologies develop, that continues to help advance the science of genomics. Couple other dates to mention here, 1990, this is when the Human Genome Project officially began. Okay, so it began in 1990, and we'll talk about the history and the timeline and the goals in a few minutes here, but it began in 1990. It was supposed to take 15 years. By 1999, the project had actually sequenced chromosome 22, number 22, not 22 chromosomes. They sequenced, the first chromosome to be sequenced was number 22. This is the general information they found. So they published in Nature, it's a scientific journal, they published in that journal December 2nd, 1999. Here's the genome information about chromosome 22. It's 33.4 million base pairs long. They found 545 genes. Expected more. They had some gaps. The technical issues were still present back in the 90s. And then they had 27 different human disorders associated with chromosome 22. They're assuming there's more, but this was the first chromosome to be sequenced. So the interesting thing, this project was geared to be a 15-year project. It took them 9 years to get one chromosome done. Everybody was looking at it and going, oh, you're never going to finish on time. It was funded by the federal government, tax dollars, Department of Energy, $3 billion. And people were so skeptical. $3 billion, 15 years, and it took you 9 years to do one chromosome? Never going to happen. But the key, the project people, 2 years later, they had the first draft of the human genome completed. The key was establishing the pattern, the template, getting it figured out. Once they figured it out, by 99, it took them 9 years to figure it out. Once they figured it out, they banged out all the other chromosomes in the next 2 years. Officially, the first phase of the project was completed in 2003. So they were under time. They were estimating 15 years. They were thinking 2005 is when they would finish. They actually got done 2 years ahead of schedule. So it was a huge accomplishment, phenomenal accomplishment for the complexity of the project. Okay? But when we look at the project, started in 90, sponsored by DOE, or Department of Energy, $3 billion of federal funding. Taxes. So $3 billion to run the genome project. That's a lot of money to go to Congress for and ask, say, hey, we need $3 billion to figure this out. So in order to get that funding, just like anything, if you're asking for money, you need to have a clear set of goals, a clear set of objectives. What are you trying to accomplish here? What is your goal or goals? What are your objectives? What's your timeline? How are you going to use the money? So fortunately, the Genome Project folks got that $3 billion of federal funding and were able to start the Genome Project in 1990. $3 billion. So if you break it down, we have 3 billion base pairs of DNA in our genetic code. It costs $1 a base pair to figure it out. Okay, so let's take a look at the goals. This is the important part. This is where it gets very interesting. Many of the goals were achieved. Some are still ongoing. They weren't goals that were going to be accomplished immediately, but one of the first goals that was laid out was that the genome founders wanted to create a physical and genetic map of the genome. Okay, so we have these. They're done. Here's the map. Here's the 23 pairs of chromosomes. Here's the physical image. How big is each chromosome? How many base pairs exist there? What information do we have available to us now? So that goal was, for the most part, accomplished and achieved. So we're all very proud of that accomplishment. Second goal, can they determine the base pair sequence in the genome? What is the first letter in the genome? What is the second letter? What is the third letter? So when you're on chromosome 7, what is the first nucleotide that you're looking at? And then the next one, the next one, the next one. A lot of that has been accomplished. That goal ties together with determining the genetic variations between Homo sapiens. And let me italicize, because anytime we do scientific names, we italicize. So the third goal and the second goal kind of go hand in hand. They wanted to look at the base pair sequencing in the genome, and then with that information, try to understand and determine genetic differences between each of us. So the way that one was tackled, they grabbed DNA from different populations, different, let's call them ethnic or geographical groups of people, and would sequence their genome. It wasn't so much between Tom versus Bill, it was more looking at the genetic variation between Europeans versus Africans versus Asians versus Native Americans and Pacific Islanders, etc. Trying to understand what are the genetic variations that exist within the human population. So a lot of that's been accomplished as well. That one, though, becomes a very sticky possible social issue that we'll talk about in the next lecture. Next goal. Can they determine the genome sequence of model organisms? Model organisms. What is the genome sequence of the mouse, of the dog, of the fruit fly, of the corn, of the E. coli bacteria, etc., etc., etc.? Knowing that helps us understand relatedness, helps us understand diversity, helps us understand evolution, helps us understand all these connections between life on Earth. And this is a big challenge. They also wanted to study the ethical, moral, and legal implications of this information. That is a huge one that definitely everybody should think about and consider. What are the possible ethical, moral, legal implications of understanding this genetic information about people, about species, etc.? Do we want to craft laws that work with our understanding of the genetic information? Do we create healthcare policies? Do we create different aspects of our lives based upon the genome information? So that's still an ongoing thing, and I'll show you some examples of how that goal there was accomplished and how that changes our society a bit here a little bit in the future, or is changing our society as we speak. Okay, so let's take a look at relatedness. This is one of the fun and exciting things about this knowledge and information. DNA relatedness to humans. Here's our next piece of information. This is yeast, common bread yeast, stuff we use to make bread, to make beer, to make wine, to make cheeses, etc. When we compare the genome of the yeast to the genome of humans, what we see is a 31% similarity. All right? That's not that close. We all understand that's not that close. But yeast is a fungi. The only connection yeast has with us is that we're in the same domain eukarya. Over here, though, we have a thing called a roundworm. Now we're in the same kingdom Animalia. And we have a 40% relatedness with that roundworm. Still not that close. It's very, very different than all of us humans. But 40%. Let's drop down to the fruit fly. The fruit fly actually is one of our model organisms. We use that for a lot of research. Thomas Hunt Morgan did some basic research with the fruit flies to determine sex linkage. Fruit flies have a 60% relatedness to us. 60% of their genome matches up and is comparable to humans. So there's a lot of similarities there that we want to try to understand and work with and gain greater knowledge about. Let's jump into mammals. Same domain, kingdom, phylum, same class. The mouse, 90% relatedness to us. So we'll talk about this more in our next lecture, looking at what significance does this have, looking at a 90% relatedness to us. Why are mice model organisms?